Hi, my name is Noor, and I work with Dr. Kochunov at the University of Maryland. Today I'm here to show you how to perform a GWAS analysis using solar eclipse software.
GWAS stands for Genome-Wide Association Study. It measures the variance in a trait that is explained by individual differences in single nucleotide polymorphisms, or SNPs. GWAS analyses are often performed to study genetic differences between two groups, such as patients and controls. The GWAS analyses I will be talking about use quantitative traits such as BMI or integrity of cerebral white matter rather than binary traits such as diagnosis. SNPs are a mutation that study a single nucleotide in a codon, allowing for genetic variance. An SNP causes genotypic variations that result in differing phenotypic expressions. These genotypic variations are called alleles and are often denoted with a letter such as A or B. It is convenient to define the major and minor alleles based on how often they occur in a population and give them numerical values. Ranks. An allele frequency measures the percentage of chromosomes that are carrying the specified allele within the population. We give the minor allele, an allele that is less frequent, for example AA, the rank of 0. We give the major allele BB the rank of 2. The genetic state that is a mixture of the minor and major alleles AB is given the rank of 1. GWAS analyses will identify SNPs that contribute to the variance in a trait. The allelic rank data is used in an additive effects model to explain the effect sizes per allelic copy. An additive effects model looks at the additive effects of each copy of an allele on the observed expression of a trait. This effect is termed beta, also known as a slope. GWAS measures beta terms for each SNP, and a small beta size generally corresponds to an insignificant association between the SNP and trait of interest. These beta terms are then used in a linear regression model to estimate the effects of a single allelic copy in the following equation. Genetic effect models include a dominant effects model in which one allele dominates the other, a recessive effects model in which the recessive allele is dominated by the other, an overdominant effect model where the heterozygous allele pair has an effect size greater than that of the homozygote, and the genotypic model where all three non-additive effects are combined for a more complex look. As you can see on this graph, the additive model reliably ranks the alleles from 0 to 1 to 2. However, significant GWAS findings can also be observed for the dominant and recessive effects. Therefore, it is always important to examine your significant SNPs for linearity of the additive effect. Here, we can see the difference in regression fit between the additive and dominance models. Both the models will produce a significant beta, however, the additive model will have a better fit to the observed data points, and therefore smaller standard error and higher significance than the dominance model. There are several ways to do a GWAS and solar eclipse that I will be going through. The choice of this approach depends on how quickly you want your results and at what precision. I will start with explaining the iterative maximum likelihood approach. This approach goes through the list of SNPs and calculates additive genetic variance in the presence and absence of the variance carried out by the SNP. Iterative maximum likelihood approach is the most appropriate way to perform a GWAS analysis. Unfortunately, it requires multiple iterative calculations of the likelihood, and therefore it is slow. It may take up to 30 seconds to perform this analysis for each SNP. It may take even longer if you use an empirical pedigree. For these practical reasons, we recommend this method to be used only for a small number of SNPs, for instance, if you only have 1,000 SNPs. This method is considered the gold standard approach. We suggest that you use this method to verify the associations from the top SNPs obtained from other methods. For MGA analysis, you need to load your trait and SNP files together from CSV files. You will need to give a prefix or SNP underscore to all the columns that contain genotype information, such as SNP underscore RS38426. Let's first start with our Plink files. We assume that the Plink file was filtered. If you do not know how to filter your Plink files, please see the video on the empirical pedigree on our website. Next, we will convert the Plink file to a set of SNPs saved in the CSV format. So we're going to use the Plink converter command to convert our Plink data into CSV data. And you will put in the command like so. You're going to put in your input file, which in my case is the external BIM, FAM, and bed files, you'll only need to put in the base file name, so without the extension, and then the output file name, the base file name that you want, so I will name mine's Geno file. Then you want to put a maximum of the number of SNPs that you have in your files. So in my case, I'll put down 3000 SNPs, and then these arguments will ensure that they are in the proper format. Once you have your Geno files created, you will input it into the snp-covar command to generate a GCOV file, which will be the source of allelic data in your MGA analysis. I want to stress, however, that your data should be accurate. If you have twins, they should have identical genotypes, or the SNP command will not run. Once you have checked your data, run the snp-covar command by inputting load snp and then your file name. In my case, I will go with this file. Now we are going to input snp-covar to form our GCOV file, and if you do not have your haplotypes, which you can do by running a separate protocol called symwalk2, then you should input the argument no haplos. Once we have our SNP data generated, we're going to run the MGA analysis by first loading our phenotype and then we're going to choose our traits. And now we are going to run MGA analysis by writing MGA and then the file that we generated for our GCOV file. And it should be the same for everyone, so and then our output files, in my case I will have mga.csv, you can have mga.out, whatever you prefer. And here we have our MGA analysis running. This command has further options that are available here under the command mga. The output of this analysis includes the number of available individuals that were tested for the specific SNP, the chi-squared test value, the p-value noting the significance of the SNP's association to the trait of interest, the beta value of that SNP, and its standard error. It also includes the extent to which the variation in the model used is accounted for, the estimated minor allele frequencies and copies per SNPs, and the standard deviation of the SNP dosage variable. The MGA approach is very slow, and therefore we suggest that you use the GWAS command. The GWAS command uses an approximation approach that will provide very accurate results, is fast, and can be used with graphics processing unit computing. However, today I will only speak about the CPU version of the GWAS command. Please see the video where I talk about the setup for solar eclipse for GPU computing. You can carry out a GWAS analysis on either a single trait or multiple traits in parallel. There are 3 ways to do a GWAS analysis for a single trait. To run the analysis, however, you need to be able to read the SNP ranking for each individual into the command, which you can do one of 2 ways. The first way to do this is by reading in the plink files .bed, .bim, and .fam, which have the SNP data by adding the argument -plink and the base file name, like this, in my case external, which you will use in conjunction with the GWAS command. Or, if you have the SNP data in your phenotype file, you do not have to add any additional arguments. However, if you have the SNP data in a plink file and would like to append it to your phenotype file, you can do so through the join command. First, we'll have to convert the plink file to a CSV file like we did earlier by typing in plink converter -i, the input file name, in our case external, and the output file. In this case, I'm going to choose append to be able to differentiate it from our other files. Then we're going to add in the argument -bin so that all of the genotyped SNP data can be converted to allelic rankings in 0s, 1s, and 2s that I spoke of earlier. And we will have a maximum number of SNPs per file. The recommended number is 5,000. Now remember that reading in the Plink files separately within their Plink format is recommended for larger datasets because it is faster. To do the analysis per chromosome, simply add in the argument -per chromo to the end of the command. So now that our files have been converted from Plink to CSV, we can append the data to our phenotype using this command. Join files out, and we're going to write the name of our output file, which will then be the phenotype file that we use for the rest of our GWAS analysis. So for example, I would write join.csv, and the first file we want to join the data from, so in this case, whatever append file, for example, append_0.csv, and then it would be the file that we want to connect it to, so in this case, the name of my phenotype file, which would be this. Now, because I already have my SNP data appended to my phenotype file, I'm not going to go ahead and do this step, but you may do so. We are now going to move on to the single trait analysis. So the first method is going to be the default method, which uses the maximum likelihood estimate. First, we're going to load our phenotype file. And then we are going to choose our traits. And then we are going to input GWAS. And then again, if you're reading in your Plink files, instead of having it pre-appended, then you would write Plink and your base file name. If not, if it's already appended, you can just write GWAS and run. Now, this method takes a good amount of time, so I'm not going to go through and wait for the entire GWAS. I'm going to go ahead and move on to the second way of doing this. So the second method also runs a maximum likelihood estimate, but it is an imputation of the SNP values with means, which allows it to run faster than the first method. You can do it this way. So first we're going to, again, load our phenotypes. And then we're going to choose our trait. And then we are going to input the command GWAS-F. And this is a pretty quick analysis. And the results are stored in a folder that is created named with whatever trait you chose from your phenotype file, and the file is inside and it will be named GWAS.out. So if you go to average DTIFA, we have all of these files and our analysis is going to be stored in GWAS.out. Here we have it, all of our results. Now, if we now want to do the third method, which only outputs the p-values from the chi-square test instead of all of the other results, we're going to do it by inputting the following. We're going to have the number of permutations. In this case, let us choose 5,000, which is the recommended number. And it will go ahead and do the analysis. This is also a pretty fast method. And again, we are going to go to our folder. And now if you look at GWAS.out, we can see that it only has the p-values. And there you have it. That is how to do CPU-based GWAS analyses for single traits with solar eclipse. Thank you for watching!